Lister share. Spore. Under suitable conditions, bacteria grow and reproduce quickly. When the environment deteriorates, such as high-temperature drying, or loss of food sources, some bacteria can use a trick to generate spores inside cells. Spores have the characteristics of dormancy and strong stress resistance, and can survive in extreme environments for decades. Therefore, spores are often used as target microorganisms in sterilization monitoring as biological indicators. So how are spores formed? Next, let's learn about the process of spore formation together. Sporulation is triggered by nutrient limitation. At the onset of sporulation, a single rod-shaped bacterial cell, termed the predivisional sporangium, divides asymmetrically, forming two cells, the small forespore, destined to become the spore, and large mother cell, nurtures the forespore. Initially, two cells lie side by side. Subsequently, the mother cell swallows the forespore in a process called engulfment that is similar to phagocytosis. During engulfment, the mother cell membranes migrate around the forespore and fuse, pinching it off as a cell within a cell. Finally, the mother cell lyses to release the mature spore. The process of bacteria turning into spores is a very interesting and mysterious process. Let's take a look together. The first key step, asymmetric cell division. Bacteria divide through the action of the tubulin-like protein FTSC. During vegetative growth, the Z-ring forms in the middle of the rod-shaped bacterial cell, marking the future site of cell division and resulting in two equal daughter cells. During sporulation, two Z-rings form, one near each pole. Only one Z-ring is converted into a division septum, the other is disassembled. The result is two daughter cells of unequal size. The second key step, chromosome segregation. The growth cells before division have two complete chromosomes, shaped somewhat like a binary star, with the DNA replication origin wrapped in the middle. In order to ensure that the forespore and mother cell each inherit a complete chromosome, when the cells start the sporulation process, the two chromosomes are rearranged into an elongated structure termed the axial filament, with the two origins of replication anchored at opposite poles. The proteins DIVIVA and RACA are important for positioning of the origins at the cell poles. DIVIVA is at the opposite poles of the cell. Here are the two DNA masses. When RACA appears, it binds at diverse sites around the two chromosomes to help collapse it, but forms a structure with many RACA molecules at the origin regions. And as depicted in this cartoon, the chromosomes get stretched out and adhere to the poles. Given this positioning of the chromosomes, only a fraction of the forespore chromosome is present in the forespore itself following asymmetric septation. The remainder of the chromosome is pumped from the mother cell into the forespore through the septum. An ADP-dependent DNA translocase located in the division septum mediates this process. The third key step is engulfment. We will also to review everything that we have talked so far. In the very first stage of asymmetric division, the Z protein is remodeled to form rings at each pole of the sporangium. Then, one of these two Z rings is converted into a division septum and the other Z ring is disassembled. While the Z rings are forming, the two chromosomes are remodeled into an axial filament by the RACA protein, and anchors the origins. Then the DNA translocase located in the division septum pumps the remainder of the chromosome into the small chamber of the sporangium, so that we have two cells that each has a complete chromosome. Next, the mother cell membrane migrates around the forespore until fully engulf it. And you will see the forespore chromosome being remodeled into a donut. The white area is the cortex, and the coat is created on the outside. Then, matures into a spore, a golf ball-like spore. The mother cell lyses and liberates the mature spore. At a moment's notice, when good condition return, it can crack open like an egg, and give rise to a cell that can resume vegetative growth and binary fission. After understanding the process of spore formation, how do spores achieve strong stress resistance? Let's continue exploring. Firstly, let's take a look at the structure of spores. This is a schematic cross-sectional diagram of a spore, and the structures from the outside to the inside are as follows: crust, coat, outer membrane, cortex, germ cell wall, inner membrane, core. Spores are killed by damage to a number of different components, including DNA, key proteins, inner membrane, and likely other components as well. DNA is the genetic material located inside the core of the spore, and once DNA is fatally damaged, the spore will completely die. The key proteins mainly refer to the enzymes involved in the main metabolic activities of spores. Once these enzymes are missing, spores cannot germinate, or even if they germinate, they cannot continue to grow. For example, corticolytic enzymes. Once corticolytic enzymes are destroyed, even if the external nutrition is sufficient, the cortex cannot decompose, and spores cannot germinate. The inner membrane wraps around the spore core and is composed of phospholipids and fatty acids. The characteristic of the inner membrane is that it has very low permeability to small uncharged molecules, even including water. The low permeability of the spore inner membrane is crucial for spores to resist toxic chemicals and maintain the stability of the core environment. Once the inner membrane is damaged, spores cannot complete germination or die directly. Given the importance of protecting spore DNA, key proteins, and inner membrane from damage for spores to survive, it is not surprising that some novel spore features help protect them. 1. Core low moisture content. 2. Saturation of DNA by alpha/beta type small molecule acid-soluble spore protein, SASP. 3. Cortical, SP, lyase binds to S-adenosylmethionine cofactor, blue, on iron-sulfur clusters, yellow and orange. 4. Pyridine 2,6-dicarboxylic acid, DPA, chelates with Ca2+ ions in spore core. 5. Low permeability of inner membrane. And so on. So how do spores use these features to protect themselves? There have been numerous studies on the resistance of spores to moist heat. Let's take a look together. In the process of moist heat sterilization, steam encounters the sterilized object, condenses, converts from a gaseous state to a liquid state, and releases enormous heat to kill microorganisms. When resisting moist heat, alpha-beta type small molecule acid-soluble spore protein, SASP, saturates DNA and provides strong protection for spore DNA, to the extent that spores die from other effects of moist heat before any DNA damage occurs. Numerous studies have also shown that the low moisture content of spore cores is crucial in resisting moist heat, with the core moisture content of spores being only 25 to 50% of that of growing cells. There are two key steps for spores to reach low moisture content. One is that during the formation of spores, the cortex expands, squeezing a rigid coat and exerting pressure on the spore core, squeezing out the water in the core. The second step is that spores absorb DPA cationic conjugates from the mother cell during the formation process, displacing water, which is commonly referred to as core mineralization. The above protective features have also been validated in our Lister laboratory. Through extensive experiments, we have found that when nutrients are restricted for about B hours, the spore is engulfed. During the CD time period, small molecule acid-soluble proteins are formed. During the DE time period, D cations accumulate significantly. During the DF time period, the spore cortex is formed. Our test results have shown that spores obtained during the EF time period obtain complete moisture heat resistance, indicating that the formation of above features enables spores to resist moisture and heat. This is the spore knowledge shared by Lister. Lister Health shares, share health.